So today I'm going to talk to you about an exploratory study that we conducted in the MiSBIE cohort, where we examined psychobiological and metabolic correlates of time perception in humans. So to begin with, what do I mean by time perception? Time perception refers to the subjective experience and the awareness of passage of time. It's a fundamental cognitive process, and that is why we are interested in it, because it's a task that it is something that affects our day-to-day life. It shapes how we perform everyday tasks, like whether it's doing simple arithmetic or following steps, while cooking. People also vary very widely on how fast or slow time feels to them, and understanding this variation in biological factors that may influence it was the central motivation for this study. And in this cohort, we use a verbal time perception task. There are lots of time perception tasks that I use. We followed a verbal time perception task that allows us to estimate both how accurate and how consistent each participant's internal clock is relative to the objective clock time. So to explain how we interpret these measures, here is a basic framework of internal clock speed. Imagine that subjective time is tracked through pulses. When internal clock is slow, the distance between the successive pulses is larger. In other words, pulses arrive more sparsely. As a result, fewer pulses accumulate during a given real-world interval. In verbal time estimation task, this makes intervals feel shorter than they really are. You can see that illustrated here with each pulse depicted as a dot, which is like spaced widely. And same sparse pulses influence the time production task as well, since it takes longer to accumulate the number of pulses that represent the target duration. People with slow clocks tend to produce longer intervals than required. When the internal clock is faster, everything is reversed. The distance between pulses is smaller, pulses arrive in rapid succession, more pulses accumulate during the same real-world interval. In verbal time estimation tasks, this makes people overestimate the duration because interval feels more filled with pulses. In time production tasks, they reach the internal target pulse count sooner, so they produce intervals which are shorter than the instructed duration. Basically, both the tasks show same directional pattern, which help us validate internal clock for interpretation. So they can be really fast clock speeds and really slow clock speeds as well. And here, each line represents a MiSBIE participant. One thing that immediately stands out is that variability increases with longer durations. When participants are asked to estimate longer intervals, their estimate becomes much more spread out. We see the exact same pattern in the production task as well. Longer target duration lead to more variable production. This is a very well-studied property called the called the scalar variability, and this also happens in our cohort. Therefore, it shows that our data was showing expected patterns of behavior. So time perception varies significantly between different individuals, and we see these beautiful intra-individual variation in prospective time in MiSBIE participants. People range from extremely fast to extremely slow clocks. For example, one participant perceived a 10-second interval as 20 seconds, whereas another participant at the opposite end perceived a 30-second interval only as 3 seconds. So the variation across trials also differed, and our most variable participant estimated the same 10-second interval anywhere from between 3 seconds to 20 seconds, and our least variable participant stayed between 8 and 11 seconds. So overall, time perception differs widely across individuals and is a very individualistic thing to be studied. So given this large behavioral variation, our next goal was to find which physiological processes shape internal clock speed and timing variability. We examined a variety of biological and psychological factors, including heart rate, age, resting energy expenditure, norepinephrine as a stress-related hormone, and immune metabolic profiles, which Cynthia spoke about in the last talk. So we first looked at, sorry, am I too close to the mic? Maybe. We first looked at norepinephrine. Serum norepinephrine was negatively correlated with clock speed, meaning that higher norepinephrine was associated with a slower internal clock. Urine norepinephrine, on the other hand, was positively correlated with timing variability, meaning that higher norepinephrine was associated with greater variability in perceived time. These relationships highlight norepinephrine as an interesting modulator, and this is something which was previously known in literature as well. We also examined age, heart rate, and resting energy expenditure. While associations were modest, there were some patterns that suggest that basic physiological state may attribute to subjective timing. Next, we looked at mitochondrial biology. We examined mitochondrial DNA content in blood cells and did not find any significant associations with clock speed or variation. We also looked at mitochondrial health index across immune cell population. Again, we did not observe overall associations with clock speed or variation except in monocytes. In monocytes, higher mitochondrial health index was associated with faster clock speed and lower variation. This suggests that immune cell types may relate differently to subjective times. Oops, sorry. Yeah. So that's what that shows. So several physiological processes are related with time perception across individuals. We also wanted to see if there were any sex-specific differences in time perception. We did not observe any differences by sex, either in clock speed or in variation. It was similarly distributed in males and females. Next was the major question where we wanted to ask whether individuals with mitochondrial disease showed altered time perception to increase the sample size. We combined all the participants with mitochondrial disease, including those with deletions, mutation, into one single mitoD cohort. And with this combined group, we found no differences between mitoD participants and controls in clock speed. Overall distributions were similar, indicating that mitochondrial disease in general does not shift baseline subjective timing. Again, with this combined group, we found no differences in variation as well. So we don't see any differences between mitoD participants and control with this particular task. However, this does not mean that the physiology of time perception relationships are same in both groups. The next question we asked was whether the correlates of time perception are different between mitoD participants and controls. For example, when we looked at resting energy expenditure, we found clear associations in control. Higher resting energy expenditure was associated with faster clock speeds, and higher resting energy expenditure was associated with lower variation. In mitoD participants, these relationships were absent. So this suggests that while average timing behavior is similar, physiological processes that may shape time perception may be different in mitoD. Next, we then examined whether peripheral immune cell bioenergetics showed similar group-specific differences in associations. In controls, higher mitochondrial DNA content in neutrophils was associated with slower clock speed and also with greater variation. Neither of these patterns were present in mitoD participants. Again, so pointing us to the fact that the group difference does not lie in the average time perception, but the underlying physiological associations. Next, we wanted to ask which neurological and psychosocial factors contribute to individual differences in clock speed and variation. Clock speed itself was not associated with any major psychosocial neurological measures. However, variation was related to fatigue, memory complaints, anxiety, and perceived efficiency. Variation in subjective time therefore captures the aspect of day-to-day cognitive function that are more subjectively noticeable. Because of the variation-related memory complaints and because we know that working memory is impacted in my mitoD patients, we next examine the relationship between working memory and time perception. Working memory supports many everyday tasks like remembering a phone number, doing mental arithmetic, reading a text, etc. And it comprises attention, memory, executive control, and the perception of time as well. So what we saw was that in controls, faster clock speed was associated with better working memory performance, and mitoD participants did not show this relationship. Again, this indicates that relationships between cognitive processes and timing differ between groups even when their average performance did not. Because of the variation-related memory complaints, we next examined the relationship, given these working memory-related findings, we examined whether intrinsic coherence within resting state brain networks relates differently to time perception. We did not observe any group differences in these associations. This was done in collaboration with Kerr and Tor, from whom you heard before. And so we found that intrinsic coherence in different brain networks were not associated with differences in time perception. We then wanted to move and finally ask if there are structural brain differences that relate differently to time perception. Here we found group differences between MITOD participants and controls. On an average, my MITOD participants had lower average cortical thickness and lower gray matter volume compared to controls. In controls, lower gray matter volume was associated with greater variation in subjective time. In MITOD participants, this association was absent. This suggests that structural brain correlates of subjective time perception were altered in mitochondrial disease. So, I feel like I've run on a breath, but what do we learn about human health from here? So time perception, first thing that I learned was that it's a deeply individual process. Internal clock speeds and timing variation differ widely across people, even within the healthy participants. And multiple psychobiological and metabolic factors are linked to differences in how individuals experience time. And the way these factors operate, they are not universal. Given that we see differences between MITOD and controls, it suggests that health conditions can shift how internal signals influence subjective time. And what do we learn about MITOD participants from here? The first thing we learn is that the pattern of influence differs from people without mitochondrial disorders. The next thing that we learn is that having a mitochondrial disorder does not automatically alter baseline time perception. So in MITOD population, again, is also very heterogeneous and they are not uniform in their timing behavior. So these are the things, these are the important factors that we learn about both these populations from this study. I would like to thank the time perception team, Gabriel, who thought of doing time perception in MiSBIE, and Catherine, Kerr, Cynthia, Tor, Michio, Carolyn, and Martin for their input in picking out what things would be interesting to test. And MiSBIE study team and participants, the funders, and also the wonderful Picard lab who have made me feel very welcome in my postdoc. Thank you so much.